Thank you, Monica. And I was telling someone this morning, I woke up this morning with sort of a panicky feeling as though my entire talk was irrelevant after hearing all of the nice discussion that we had yesterday about the genomics-driven clinical trials and drug discovery. But when I thought about it some more, I realized that while those are absolutely wonderful examples, and I hope where we're going to be in another few years, that we still have a lot of other work to do in oncology. And it's not all going to be the miraculous stories of give a patient a drug based on their mutation and you have a miraculous cure. So in that spirit, I will go through with my slides. And what I'm really going to try and do is give you some of the day-to-day challenges that we face as we are dealing with the world of biomarker-driven trials and even other kinds of trials that we could do in this new and wonderful cooperative group system that we have, this National Trials Network. Because there were some examples mentioned even yesterday of studies that maybe are not the fancy genomic-based studies that really still need to be done and could perhaps be done nowhere better than in our National Trials Network. So whenever I'm requested to give a talk on innovative trial design, I always like to remind myself and remind the audience that innovation is great, but we have to make sure that the innovative methods still answer the questions that need to be answered. And so I sort of came up with this following outline for my talk, which is that first I want to review the goals of statistical design. I'll talk a little bit about some different design approaches, and then give you a couple of cautions and caveats, and then sum up with some thoughts. So our number one goal with designing a clinical trial should be that we want to end up with interpretable results. Okay? And ideally, we would like to do that in such a way as to be most efficient with resources. We'd like to do it with the least number of patients and do it in the least amount of time. And it'd be really nice if, in addition to answering the question we set out to answer, we have learned new information that might help us in planning future trials. It struck me as I was beginning to think about this talk that there's this sharp contrast in the landscape as it reflects what's happening with the National Trials Network and what's happening with the way we are approaching oncology trials. For the National Trials Network, we have reduced the number of cooperative groups, but each group is larger, more diverse, and we also want to increase the capability for those outside of the groups to enter patients onto the trials. In oncology, we're sort of going the opposite direction, that in oncology, we're dividing our cancer types into finer and finer subgroups, and that we're developing therapies that are targeting these very rare molecularly defined subgroups. We do, however, have expectations for bigger treatment effects in those smaller subgroups. And so how do we deal with this new paradigm statistically? So some of the designs I'm going to talk about, and there's only so much I can do in 20 minutes, but I do want to focus on some issues in biomarker-based clinical trial design, talk about some things that would apply more generally than biomarker-based trials, things like multi-arm trials, some adaptive features, you hear that term a lot, and also maybe some cautions with regard to use of intermediate endpoints, which is one of the ways in which we think we can be innovative with trials and perhaps speed up the rate of discovering and approving new drugs. So for biomarker-based trial designs, I lump them into roughly 4 categories, and you can have hybrids of these categories. And I'll go through each of these in turn, so I'm not going to read them here. But it's noteworthy that one of the first papers that really sort of laid out the framework for biomarker-based clinical trial designs was really only published in 2005. While people might have been sort of doing this informally for a while, it was Dan Sargent and colleagues, some of whom are in this room, like Rich Shilsky, who really came up with the basic framework to how to start thinking about using biomarkers in clinical trials. So the design that we're tempted to do whenever we have a biomarker that we think is related to response to a therapy or benefit from a therapy is something called the biomarker enrichment design. And so, for example, the development of Herceptin and companion diagnostic Hercept test really followed this design. And the simple idea is that if you think that your drug is likely to benefit patients who have a particular marker, In the case of Herceptin, it was HER2. That maybe it's only those patients who you want to actually randomize. So there was a clinical trials grade assay in the development of Herceptin, and they used that to screen patients for those who were positive for the marker, and they randomized to the new agent, namely what became Herceptin, to control therapy initially in the advanced disease setting. So you might ask first, well, why did we actually need to have a control therapy? Why did we need a randomization? Well, one of the reasons is that some of these markers, in addition to predicting who is likely to benefit from the drug, may be prognostic. And in fact, in the case of HER2, it's actually negatively prognostic, meaning you tend to do worse if you are HER2 positive, if you get standard therapies. So that's one realization, first of all. And the other realization, though, is that when you use this kind of design, you have sort of narrowed the field. And we're still struggling with this, to be honest, in the field of HER2, that if you don't randomize the patients or don't study the patients who are negative for your marker, you never really know if you got the marker right initially. And here we are in 2013, and I've been a member of 2 CAP ASCO panels for the evaluation of HER2 status. Okay, so we're still debating this. And we have, within the last few years, started up a trial in the adjuvant setting where we are trying to determine if patients who we would have traditionally called HER2 negative by our approved assays, whether they might benefit from Herceptin as well. Okay, so it's not such a simple thing, and you need to think as you're developing these drugs and developing these markers when you want to sort of nail it down and maybe limit your opportunities for the future. And I'd also mentioned, well, I guess I said the key point there really is that the enrichment design doesn't really let you know what's going on with the marker negative patients. If you want to kind of hedge your bets, you could do a biomarker stratified design, which is that you randomize everybody, but you might stratify your randomization on the marker to ensure that you get a balance of treatment arms within each of the marker subgroups. The nice thing about this type of design is that it really leaves you a lot of options open for going back and refining the biomarker, because just because you stratified randomization on the biomarker doesn't mean you can't go back and look at another biomarker, perhaps another biomarker that's on the same pathway as what you thought was the right biomarker initially, or other kinds of refinements. So there's a lot of reasons statistically to want to do a design like this. In practical terms, it can be difficult to accrue to trials like this because people tend to grab onto biomarkers and believe them immediately. So it can be very difficult to randomize to a study in which you're going to randomize people who are marker negative to a new therapy that everyone thinks is probably targeting a marker that you don't have. Biomarker strategy design is one that we statisticians generally do not recommend. It sounds very attractive because the way it works is that we randomize you between getting the marker and using it to determine your therapy versus not having the marker assessed and just going on to whatever the control therapy is. Well, the main limitation of this design is that you can see that if you're marker negative, if you get randomized to the arm that uses the marker, you get control therapy. If you get randomized to the arm that does not use the marker, you also get the control therapy. So you're really wasting those patients. You are not getting full information out of those patients. And it also doesn't allow you to pick up other aspects like prognostic effects, et cetera. But I won't go into detail here because basically I'm saying I don't recommend this design. So we heard about some trials yesterday that in effect are just souped up versions of some of the basic trial designs that I just spoke about. We could have what I'd call a multiple biomarker signal finding design, which was basically what was referred to yesterday as a basket design. Okay, so what the basket design says is you bring in all your patients, you screen them for a series of markers. If you have marker A, you get the agent A that targets that marker. Marker B, you get agent B, et cetera. The beauty of this is really, there's nothing fancy statistically here. I mean, this is simply a series of single-arm trials where we are performing the trials in the different marker-defined subgroups. Okay, what is really nice and what is efficient or innovative here is really the logistics of running such a trial, because we have a big screening protocol that brings everybody into the same front door and then directs you to basically separate individual single-arm trials. Okay, so this is not really a statistical efficiency or a statistical innovation. It's how we do business. It's how we carry out the logistics of the trial. But we should remind ourselves that there are still questions here that should be carefully considered before we launch one of these studies. First is, what is the right endpoint? Okay. Most of these kinds of trials that we're hearing about, people are using something like a response endpoint. We could, of course, have some agents that really don't necessarily give a lot of responses. Maybe they will, put a patient into more of a stable disease state. If we're looking at something like progression-free survival as our endpoint, suppose progression-free survival, 6-month progression-free survival. Well, suppose you have an agent or let's suppose you have a marker that is associated with a more indolent course of disease. So even if it's an advanced disease state, maybe some advanced patients will progress faster than others. Well, then what do you choose for your benchmark for progression-free survival? Is 6 months the right number? Should you look for a year? What is it? So we can't assess those kinds of things in this type of design where we don't do any randomization. So we could decide to put randomization into a trial like this. And now what I'm looking at, I've called it the multiple biomarker randomized enrichment design, which is basically a bunch of enrichment designs run in parallel for each of your different marker subgroups. Okay. So this has the nice features of randomization, namely you can distinguish prognostic from predictive effects. And you can have variations on this. If you have a situation where there is a standard therapy available for the patients, you can randomize to standard therapy plus or minus the new agent instead of standard therapy versus targeted agent. What we can't do is some of the same kind of limitations that we have with the simple enrichment design is that we can't assess off-target effects. We don't know if perhaps we give agent A to somebody who's positive for marker C, that in fact maybe they'll get some benefit too. We never really stray outside of our biomarker-defined subgroups. And again, this has some of the nice logistical efficiencies as we've already talked about with regard to the basket design. So some might say that it's really difficult to synchronize the development of the biomarker and the drug, and we certainly know of examples. The anti-angiogenesis agents, for example, we really haven't come up with some really strong biomarkers there to predict who's going to benefit from those agents. So some would say that we ought to keep doing our sort of broad brush approach where we randomize all patients and then try and go back, collect the specimens and go back and figure it out later. And in fact, this can be a very valuable approach. And if you use the specimens wisely, and you have very carefully defined, pre-specified statistical analysis plans, you're careful about how you do your assays, and a number of other features, that you can still answer some important questions operating in this kind of mode. The thing I would remind you of, though, is that if what you're really looking for is markers that predict who benefits from therapy, as opposed to, for example, prognostic markers, that if you have designed a trial to answer a treatment question, it may not have sufficient sample size to answer a biomarker treatment guiding kind of question. And the reason I say that is that if you're trying to look for a biomarker that defines who does and doesn't benefit from a therapy, that you basically want to say, and keeping it simple as though we had a binary biomarker, that if you're positive for the biomarker, then there is a treatment benefit in that subgroup. But if you're negative for the biomarker, you want to be able to say with some degree of certainty that there is no sort of clinically meaningful benefit of the treatment in that biomarker negative subgroup. Well, you're now asking two questions, and you're asking those questions in smaller groups than your original trial. So you may not have the statistical power, particularly in the biomarker negative group, to rule out that there's any benefit in the biomarker negative group. Depending on the situation, you may feel as though you know the biology well enough that that's not a likely situation. But again, many times we don't know the biology that well. So in terms of practical issues, as we have had our discussions at NCI in cooperation with the cooperative groups, there's still a lot of devil in the details when you're trying to launch some of these types of trials. And some of the big ones really deal with the biomarker-based testing. And so, for example, you heard about the Alchemist trial yesterday, which is a study in the adjuvant setting in lung cancer, a collaboration with the Alliance and with ECOG, where we're doing randomized studies in the ALK positive group and in the EGFR positive group. And so one of the first questions we had was, so what do we do with the assays? So there is an FDA-approved test for ALK rearrangement. There is not currently an FDA-approved test for EGFR mutation testing, despite the fact that it's being done in labs all over the country. So what does that mean for a trial? And what does that mean for the interpretability of a trial? You know, we do have to worry about the possibility there could be extreme heterogeneity in the marker testing if we just throw it out there to any lab that wants to do it. But of course, then it costs money, and there are logistical considerations in having it all done in a central lab. And so if you do have take the all-comers kind of approach with regards to the laboratory assays, how can you be sure for which of those marker assays the results of your study actually apply? Because I would venture to say that if you open up to enough labs, you may get a couple of labs that maybe do not use the optimal method for assessing EGFR mutation status, let's say. So this is also a concern for the patients, and I'd love to hear some comments from our patient advocates in the audience later on when we get into the discussion, that how do you know as a patient that once this drug gets out there in that setting, if the trial is successful, that the lab that your sample is sent to is one that is going to do it the right way or an acceptable way. These are really big issues. Other issues, we heard a little bit about IDEs from Kim Jessup yesterday. How does one deal with an IDE in this situation? You can't possibly go out and test every single lab that's going to be involved in doing this mutation testing. So can you narrow it to a few? How do you deal with all of this? So those are some real, like I say, practical issues that we are trying to get our brains around. The other big question, again, I'd love to hear some comments from the patient advocates. What do you do with the discordant local positive and central negative cases? So if we allow for this, all-comers kind of laboratory testing, and I think we're sort of moving in the direction of at least saying, well, we're going to do central testing at least after the fact. What do you do with those patients if they're centrally negative, even though they were positive and got onto the trial? Or should we not allow them onto the trial until we have the results? And in the adjuvant setting, you can do this. You have some time. Advanced disease, you don't have unlimited time to go back and do the central testing. So what do we do with those? On the one hand, you say, well, maybe we could learn something from those discordant cases. Maybe the central test method is not the absolute best method, and maybe it's good to get that heterogeneity and see what we can figure out from that. But on the other hand, as a statistician, I say to myself, well, all right, hopefully we're not going to have a huge percentage of discordant cases. And so suppose we get a few dozen of these cases. Can we actually answer any question using the information from those cases? I can't come up with a power calculation that's going to convince you that we could actually make a definitive conclusion based on whatever we see happening in those patients who are discordant if we let them on the study. These are big questions. And also has bearing on your statistical analysis. Should the primary analysis be limited to the group who were confirmed centrally, or should it be sort of the broad group? And again, it depends partly on your feelings about, well, are we trying to project to the relevance of these trial results for the entire community, if this is the way the testing is going to be done in reality, or do we sort of want to make sure we have a more scientifically rigorous result? Okay, so that's my spiel on biomarker tests, and I'm just going to mention a couple real quickly ideas for how we might bring some more innovation into how we do studies for non-biomarker guided kinds of therapies. And a couple of things I want to mention are multi-arm trials. And here what we do is we have multiple agents tested together in the same trial. There's a statistical efficiency in that approach in that you can reuse the control arm. And this is a couple of calculations that show you how much efficiency you could gain. So how many fewer patients can you accrue to the trial to answer your scientific questions? These are kinds of trials, particularly if you're trying to look at drugs that are produced by two different drug companies. The drug companies are not going to run these trials. So if we have important questions of this type that need to be answered, the National Trials Network is an ideal place to answer them. Factorial designs is another kind of approach you can take. It's really, again, a multi-arm trial, but it's a little fancier in that the multiple arms are actually constructed from a yes/no on each of 2 treatments. And you could, of course, have more than 2 treatments as well if you wanted to. And the efficiency in this one is that you can, again, reuse patients, because to answer the agent A question, you can compare the 2 rows. To answer the agent B question, you compare the 2 columns. So statistically, there are a lot of advantages to this, although it does require an assumption that you do not have interactions between the drugs. So I think I've already pretty much mentioned the advantages and challenges in this. I am running out of time, so I can't talk a lot about adaptive designs other than to mention that there are many different ways you can do adaptive designs, and some of the things we are already doing in our trials. Interim monitoring is a type of adaptive design where you periodically check, and if you have enough information that's convincing enough, you stop the trial early. We have early slow accrual guidelines in place now. That is really another way of doing an adaptive design, sort of based on feasibility. And we can drop ineffective arms or non-benefiting patient subgroups. This can all be handled statistically. You just have to decide how you want to do it. It has to be pre-specified in the analysis plan. We're doing more and more phase 2/3 designs now. The thought is that instead of having this abrupt end at the conclusion of a phase 2 trial and then having to go through all the paperwork and get everything up and rolling again for a separate phase 3 trial, can't you take those patients who were on the phase 2 trial, simply follow them longer right on into the phase 3 trial, assuming that the phase 2 results were sort of reasonably promising, whatever that means, and that's where the big questions are, of course, that you could have this sort of seamless transition. So we have a couple of designs right now in the cooperative groups that are of this type. However, I do want to issue a caution that, again, one of the big questions in a design like that is, what is the right intermediate endpoint to base that decision upon regarding whether you want to transition from the phase 2 to the phase 3, or if you just call it quits at the phase 2. And I was struck by some data that I saw presented recently. This was presented at the San Antonio Breast Cancer Symposium. They were looking at PCR as an intermediate endpoint in breast cancer. And this is a very popular thing nowadays, to run neoadjuvant trials in breast cancer and to use pathologic complete response as an endpoint, as thinking being that this would be a useful screen for what can go on to a more definitive trial. And with the kind permission of Pat Cortazar at the FDA, she allowed me to share with you some of these slides. And they're pretty sobering. Okay, so what you're looking at here is, this is the pathologic complete response odds ratio. So it's the measure of the association between pathologic complete response and treatment. So the null value here is at 1, and here's your definitive endpoint. In this case, it was overall survival. There's your null value. There's very little association between pathologic complete response endpoint and overall survival. This should make us a little bit worried about the trials we have going on right now using PCR as an endpoint. And of course, if you're like me, you raise your hand immediately and say, well, wait a minute, we already know that an ER positive disease, pathologic complete response, is not really, kind of the right thing to use. And so I asked that question when Pat came to talk to us. And she said, well, unfortunately, if we take out the hormone receptor positive early, lower grade cases, it still doesn't look very good. Okay, so this is just a word of caution that we can be innovative, we can make assumptions, but we need to be doing some reality checks too, and make sure that what we're doing is the right thing. Okay, and anyway, just to sum up, I think that there's, I feel really encouraged based on the last 2 days of discussion, and I think that this new trials network has a lot of potential to do important things in better ways, more efficient ways than we have been doing in the past. And you have some capabilities that no other organization in the world probably has. You have the expertise, you have a system in which you can bring in a lot of patients, and I think you should all feel very optimistic about it. Thank you.
Okay, so before I introduce our last speaker of this session, I just want to sum up a little bit. Yesterday we heard about the new system. We heard about the improvements that have been made over the last 2 years. We heard about the capabilities of the new National Clinical Trials Network. And I think today, at least the theme of this particular session is, who are we to serve, here to serve? And obviously, as Bob Comas described in his slide yesterday, we view the National Clinical Trials Network as the nidus at the center of a very, very complex environment that is trying to change patient care. And it's very clear from both yesterday and today the great diversity and complexity of that enterprise. We heard today in the talks first from Ed about we are here to serve as the direct handoff to the discovery, immediate translational world, and we cannot be a silo over in our clinical trials world. We've got to understand what is going on immediately in the discovery, early translational realm. Otherwise, we won't be relevant. We heard from Renaud about how we exist if we're going to treat patients and develop new treatments in a global world now. It's no longer about the patients in the US, although our efforts are keyed in on bringing the North American, United States, and particularly the community network into this environment so that it's relevant. We've heard from Hans-Georges about how we exist in an environment where bringing data to the enterprise is important, and how that is a changing landscape, not only from year to year, but from trial to trial. We've got to maintain the flexibility and the ability to work very closely with our regulatory partners, industry partners, and discovery partners to make that happen. And then finally, Lisa was really here, and Lisa, you couldn't have given a better talk, because Lisa was here to illustrate that we are here to serve our investigators and to listen to our investigators and to be able to respond in the system with the flexibility that our investigators dictate, so that we really are getting the best and the brightest innovation into the system. So, it's a daunting task, and I think that this is what makes this such an incredibly rewarding place to be as a participant in these groups. So finally, let me introduce our last speaker, because this is the most important speaker of the session. Sorry, everyone else. Ultimately, we are here to serve the patient, absolutely. And the most important partner we have in anything that we do is the patient. And so I would like to introduce one of the most knowledgeable and valuable contributors, certainly that I have ever met, Pat Gavin, who is a representative here today from the patient advocate community. He is on the executive board and the chair of the advocate committee of the Grand Rapids Clinical Oncology Program, among many other things. So Pat, welcome.